Hi, everybody. Okay, so we finished the lecture on operons, and that's 18.1 of the chapter. And then we're continuing in the chapter about gene regulation. Now we turn to our talk about eukaryotes, which, of course, are going to be far more complex. So we're going to kind of go back and forth a little bit, but just want to tell you what we know about eukaryotes and give you some terms and kind of describe to you eukaryotic genes and what we know about them now. Okay, and we're learning a heck of a lot more. Next week, you'll be doing a activity from HHMI. You're going to see the PIDX1 gene. Okay, so this is 18.2. It says eukaryotic gene expression can be regulated at any stage. So we can regulate gene expression at the transcriptional level. That is, we prevent transcription from happening. It can happen after transcription, and that's, as you know, we can snip out the the introns, and splice together the exons. You can actually even transcribe the gene, make the mRNA, and then the mRNA is never read, never gets to the ribosome. It can be destroyed, okay, even before it gets to the ribosome. And so that's called post-transcriptional level. And it can even be done at the translational level, where an organism goes through and it transcribes the mRNA. It reads the mRNA at the ribosome and codes it, and then the protein gets destroyed. Okay, maybe it's not needed or something like that. Lots of interesting stuff here. Okay, so what's differential gene expression? Just like it says, all differential gene expression means is that every single cell in your body has the exact same copy of the genome, but only certain cells read a certain section of that genome. I make it very similar to a textbook, like your bio textbook. Everyone in your class has that textbook. But what if some people only read like the first page, and some people read the whole thing, and some people read certain sections of it? That's exactly how your cells work. Okay, they have every single cell in your body has the full complement of DNA, but certain cells will read certain sections of that DNA. Okay, so it says, with the exception of some of the immune system cells, all the cells in our bodies have the exact same genome. Most cells only express about 20% of the genome. That's probably high, whereas highly specialized cells express even less. Different types of cells are not due to having different types of genomes, but rather expressing different genes within the genome. Okay, and then I make a joke here about not reading the textbook much. Haha, very funny. Okay, another thing is the chromatin structure. Now, remember those chromosomes. Sorry about that. There are times when the chromosomes are super, super, super tightly wound. That's typically going into mitosis, and it's actually called heterochromatin. And there's times when the chromatin is super, super loose, all right? And that's called euchromatin. You don't really need to know those types of chromatin, but I like to bring it up. So the difference is this: when chromatin is wound super, super, super tight, all those transcription factors that get into the DNA and they attach to the DNA and they attract RNA polymerase cannot. So therefore, you don't read the DNA. Whereas when the chromatin is real loose, okay, the transcription factors can get in there. They can bind to the DNA and they can attract RNA polymerase. And as a result, whenever RNA polymerase attaches to something, it's going to read it. So you do read the genes in that situation. It's funny because if you know anything about race car driving, one thing about race cars is if you keep your suspension loose, the car is real, real fast. It's hard to control, but it's real fast. And I kind of remember it that way. Whereas with the car, when the car is tight, when you make the suspension super, super tight, it's easy to control, but the car is not real fast in that situation. So both bike riding and car racing had something going on there. Okay, so what winds up DNA? Well, there's got to be a reason or a way that it's done. And the big one is called methylation, okay? But when you attach methyl groups, so I'll go to the second one first here. When you attach methyl groups to DNA, all right, you remember your old functional group methyl groups, they're nonpolar. That causes the DNA to wind up tight. And because it winds up tight, the DNA is not read, all right? In bacteria, when a virus gets into that bacteria, the way that they can survive from that virus is they methylate their own bacteria. And by doing so, they wind their DNA up super tight, and the viral DNA can't get inserted into theirs. Okay. And then there's two other methods that do the reverse. All right. And that's called acetylation, attaching an acetyl group, COOH, or phosphorylating DNA. Both typically activate it. Both typically loosen it up. But the most famous, and one that you're going to want to remember, is the methylation. Methylation, usually 99% of the time, causes DNA to turn off. Very, very rarely, on very, very rare cases, will it turn on. And so once this was discovered, we're getting into something called epigenetics. And epigenetics is the understanding that organisms can carry genes, but they can also carry genes that are turned off. And as a result, they pass these genes on to their offspring, and even the offspring have the genes, but they may pass the genes turned off. And as a result, the offspring do not express it. So epigenetics is this idea that there's another layer on your DNA. It's one thing to have the code, it's another thing to express the code. So think of it like a switch. And epigenetics is literally the idea that there are switches that control your DNA. And a lot of research came out on this stuff in the late 80s and in particular in the 90s. Okay, so just remember methylation, it's going to wind that DNA up tight. So I just wrote again in your notes for you, DNA methylation of DNA can occur at the histone tail level or the nitrogenous base level. So those methyl groups can attach to the histones, those things that the DNA winds around, or can attach to the actual nitrogenous bases. Doesn't matter, okay? And different enzymes can methylate, but methylation usually means turn off, all right? Another example of methylation is like the bar bodies, if you remember those in females, how they have a dark staining body inside their cells because every single individual has at least one, just one X chromosome that's going to be active, and the other one is inactive. And you guessed it, that inactive X chromosome is heavily methylated right here. Okay, so you can just read along in your notes, and I'm just kind of giving you the overview, and you can just kind of read along in your notes. Genomic imprinting. So genomic imprinting is when organisms, mom or dad, pass on genes to their children, but those genes that they pass on are heavily methylated, which means they're turned off. So it's more or less where the methylated genes are passed down from parent to offspring. So here was a quick experiment that was done with agouti mice. All right, and what they found was that it wasn't that these parents were passing on, not passing on genes to their offspring. It's that they were passing on methylated genes. So in the control group, the pregnant mothers were given a unhealthy diet, and when they did that, the babies, these are mice, the babies were born with a yellow coat, and they became fat. Okay, whereas the experimental group, the pregnant mothers were given a healthy diet, okay, with supplements, and the babies were born with a brown coat color. Okay, these are called agouti mice, and they stayed thin. Okay, so this was a lot about the fact that these moms were passing on methylated DNA to their babies. Okay, so the inheritance of traits is not directly tied to the nucleotide sequence, but some other mechanisms such as methylation. So learning how to silence some genes in the human body and turn on some genes in the human body are a huge part of bio now. And remember, many times we can trace disease to a gene that is not methylated and it should be, or vice versa. So I'm just trying to give you some ideas of what's called epigenetics, just this overlying uh, portion of the genome, epi meaning outer, and basically what we're dealing with here is switches, guys. That's about as much as you would need to know at this point. Okay. So in eukaryotes, regulation of transcription is much more complex uh, than prokaryotes. It's much more complex in eukaryotes than prokaryotes. Okay, so let's talk about what we know about most eukaryotic genes and their basic structure. All right, so this is basically what it looks like. And in a eukaryote, you're going to see the end of the gene, and you're going to see that genes have introns and exons. Okay, remember, based upon the individual, some people's introns are other people's exons. So this could be for Mr. Later, it could be intron, 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 exon, exon. For you guys, it could be exon, intron, exon, intron. Very, very different here. Okay, this is the promoter region, and the promoter region is where our good friend RNA polymerase is going to bind. Okay, and this is also we talked about usually a series of T's and A's, the TA box on the coding strand. On the non-coding strand, it would be AT. On the coding strand, TA. Now, there's also something called control elements. This is new for eukaryotes. Control elements, okay, and there's two types, as we're going to talk about, are non-coding DNA. Remember, out of all your DNA, about 98.5% of it is not turned into proteins. Only about 1.5% is red. So, if I look right here, not red, this is not turned into protein, this is not turned into protein, this is not turned into protein. So these are the switches, guys, and the switches are called control elements. And there's two types. There's the proximal control elements. The proximal control elements are the ones who are close to the promoter, and thus proximal meaning in close proximity. All right, so the proximal control elements are close to the promoter. And the distal ones, they're going to be very, very far. They're going to be upstream. So they can be thousands of nitrogenous bases away. And the distal control elements are the ones that are going to be further away from the promoter. Sometimes these distal control elements are called enhancer region. Sometimes even repressor region. But usually like the enhancer region and the proximals, they usually just are called control elements or proximals. So just so you know, typically the proximal control elements kind of act like the operator in a prokaryote. Remember, we're related. This is where usually general transcription factors are going to bind. And when general transcription factors bind, they attract RNA polymerase and they either turn the gene on or not. In the distals, this is usually where specific transcription factors bind. And specific transcription factors are typically either your repressor proteins or your enhancer. And based upon the name, you should get an idea of what that's going to do. The repressor proteins are going to slow it down, okay? And the enhancer proteins or activator proteins are going to speed it up. So this is like the on-off switch, and this is the dimmer portion, okay? Since Mr. Later put in a new light this weekend, I'm thinking all about dimmers because I put a lot of those in. All right, so I would have showed you pictures of my project, but not now. All right, so organization of a typical eukaryotic gene. A eukaryotic gene does not follow the Alperon model. Instead, most eukaryotic genes have the following features. They have introns and exons like I showed you. All right, a promoter region where RNA polymerase binds. That's going to be right there. Okay, that's promoter, so it goes back and forth there from both prokaryotes and eukaryotes. And control elements, segments of non-coding DNA, just non-coding DNA. They're called switches, all right, located upstream of promoter that bind to proteins and regulate transcription. These control elements and the proteins they bind are critical to the precise regulation of gene expression, okay? They're called switches. That's what they are, okay? Control elements are switches. When this book was actually written, they weren't so sure. But in the past several years, they're sure. Okay. Now remember what those transcription factors are. There's two types. All right. So there's two types of transcription factors that bind to these control elements. There's the generals. All right. And the general transcription factors, what I want you guys to know is that the generals, they're usually going to bind to areas near the TA box, the proximal control elements, and they basically get transcription started. They basically turn it on or off. And then the specific transcription factors, those are going to be called repressors or activators. And what they're going to do is either speed it up or slow it down. How do they speed it up? They attract a lot of RNA polymerase. How do they slow it down? They don't attract a lot of RNA polymerase. So I just kind of go into that right here too. The two types of control elements, what I told you, the enhancer region and the proximal region, and that is going to be it for 18.2. So after this, we have to talk about combinatorial and coordinative control of genes, and then how we kind of regulate DNA. But it gets into a lot of stuff here, and I will go through this stuff kind of fast. Okay, so for right now, we are through a good section of the chapter. Thanks for hanging in there, guys.